Right. Today we are going to talk about blood-brain barrier. Right. Now, what is blood-brain barrier? It's not something like a physical barrier as they are in the road and you cannot pass through. Blood-brain barrier is a basically concept which was originally originated in 1882 by experiments of a doctor, Paul Ehrlich. Now, what did he do? Actually, he took live animals, thank God animals, right? But even animal rights people will not be happy about it. But what did he really do? He took the live animals. Let's suppose here happens to be an animal, right? And let's suppose here is the central nervous system of the animal, right? What did he do? That he injected special type of dyes, right, in their vascular system, right? Vital dyes, vital dye, to be very exact, trypan blue, and this molecule of trypan blue, now we know that basically it binds with albumin or with the plasma proteins. Now, Dr. Paul Ehrlich, he injected the live animals, the living animals, with vital dyes, right, in their circulatory system, and then he tried to study that which organs of the animal are colored by the dye. This was a blue color. Which organs of the animal turned blue? What he really found that this injection of the blue dye, right, trypan blue, it colored almost everything in the body except central nervous system. All the tissues turned blue, right? And this blue dye approached all the tissues in the body, but it failed to enter into central nervous system. So he thought that there must be some sort of barrier between circulatory system and the central nervous system, right? And this was the origin of the concept of blood-brain barrier. This was a very primitive concept, very initial concept. That what is blood-brain barrier? That Paul Ehrlich injected basically dyes in the vascular system of the living animals, and then as these dyes were protein binding dye, and then he studied the distribution of the color in different tissues. And what did he find? That this blue dye was present in all the tissues, almost all tissues, but failed to enter into central nervous system. Then he thought that it means there is some barrier between the blood and the brain, and of course blood and the spinal cord also. And later on we discovered similar barrier is present between, not only between blood and brain, but also present between blood and spinal cord, and also between the blood and peripheral nerves. Even this dye failed to color the internal side of the internal part of the peripheral nerves. So truly now we believe this is not only blood-brain barrier. Even though in common language when doctors discuss, they talk only about blood-brain barrier. But actually when we are talking about blood-brain barrier, we are talking about three things. Blood and brain barrier, and also it's understood we are talking about blood and spinal cord barrier, and similar barrier is present between the blood and peripheral nerves. So blood and nerve barrier. Am I clear? Now the question is this. Okay, Mr. Paul Ehrlich made trouble for us that now we have to study this initial discovery. But as time went by and there were better techniques to study the, you can say, this blood-brain barrier, to look at it, what anatomically exactly it is and what structurally it is, right? Now we know a lot about it, right? Now we'll go into more advances and we'll talk about that what is exactly blood-brain barrier. What is that structure and how that structure work which prevent many substances from the blood to enter into central nervous system freely. Central nervous system is like a, what should I say? One thing you know, that central nervous system has a lot of neurons. And neurons are the master controller and regulator of the body. And neurons are very sensitive, just like ladies, right? Overly sensitive. And neurons like that approach to them should not be thoroughfare. Is that right? These neurons are like ladies, very, very sensitive and very specific. And they like to keep their private environment. And they want to keep a very stable environment. Not whatever fluctuations are going in blood should also come into their environment in the central nervous system. Right? So let's talk about that. Here is a neuron. And this neuron, this is a group of neurons here. Right? These are very, very sensitive. And everything from the blood should not reach there. There must be some barrier. Most of the women believe in barriers. Right? Sometimes they put emotional barrier, sometimes they push psychological barrier. In the same way, neurons have put a lot of barriers. We'll talk about that. But why? Why there is barrier? Why should be a barrier? The reason being that even though neurons have to get the nutrition from the blood and neurons have to release their waste product into blood, but still all fluctuations and all the drama of blood chemistry should not disturb the biochemical or microenvironment of the neurons in central nervous system. Right? For example, in the blood, hormonal fluctuation occurs very frequently. In fasting period, hormonal composition is slightly different. And postprandial, once you have eaten a lot of food, your hormonal composition in the blood is different. If you have done a lot of physical activity, your hormonal composition in the blood fluctuates. You know that about insulin, glucagon and other hormones. If you are under stress, again hormonal fluctuations occur. If a big black dog is after you, body surges a lot of norepinephrine in the blood. Is that right? So blood chemistry changes. As I told you, blood hormones change. But neurons sitting here, we can say ladies' room, right? These neurons don't love every fluctuation should come there and disturb their special functions, right? So there must be blood-brain barrier. So one of the function of the blood-brain barrier is, I will tell you later what exactly blood-brain barrier is, but one of the function of the blood-brain barrier is that it protects the extracellular environment in the neuron, in the central nervous system, the microenvironment of the neuron or biochemical environment of the neuron or extracellular environment of the neuron. It protects them from major fluctuations in the blood. And not only protect, actually it is responsible, blood-brain barrier is responsible to keep the neuronal environment stable, relatively stable. And neuronal environment should not be at the mercy of changes in the blood. Right? As I told you that if hormones are fluctuating in peripheral circulation, this should not reflect in the same degree of fluctuations in central nervous system. Right? If there are metabolites in the blood or glucose level in the blood fluctuate a lot. When you take food, amino acid level in the blood fluctuates a lot in the fasting state and in the fed state. But those fluctuations should not be that much to the central nervous system, maybe to some degree. Then metabolites in the blood with exercise, right? Their amount goes very high, and resting period it becomes less. Again, it should not bother the private and discrete world of the neurons. So one of the major function is that it is protecting the central nervous system microenvironment in which the neurons are working, right, to keep them into stable stable environment and protect central nervous system extracellular environment from the fluctuating hormonal levels, metabolites levels in the peripheral blood and even ionic levels, right? Because neurons love to have very stable level of sodium, calcium, potassium, right, magnesium, chloride, bicarbonate, protons, and there are relatively more fluctuations in the peripheral circulation. So this is one purpose. So we can say blood-brain barrier act as a what? Stabilizer. It acts as a stabilizer. It stabilizes the internal environment of central nervous system, including brain and spinal cord, right? Then it has one more function. Not only these lady-like neurons love to keep their environment relatively stable and relatively less fluctuating, they also want to be protected there. You know, ladies need more protection than men usually, even though you come across sometimes very dangerous ladies. Another function is protection. Now, central nervous system, blood-brain barrier protected from toxins, endogenous toxins or exogenous toxins, right, which are produced in the body, endogenous toxins, or if you have taken parenterally or some toxins, you have taken gastrointestinal system or some other way, right, there must be mechanism to protect the central nervous system from that. So they have one more function. For the not only stabilization, it acts as a stabilizer, blood-brain barrier, it also acts as a protector. It protected from the toxins. Is that right? Even it protected from the toxic range of the drugs. Sometimes you give drugs. All drugs don't enter into central nervous system. Selected drugs do enter, right? So protected from many, many toxic drugs. Then it protected from the microbes also. It protected from the microbe also, especially particulate microbes like bacteria, right? For bacteria, usually it's a little bit resistant to enter into central nervous system. Is that right? Then there is one more function. Not only it's stabilizer of the microenvironment in central nervous system, not only it protected from the toxic substances, endogenous like bilirubin or exogenous like neurotoxins, another thing, it's a very good holder. Holding of what? Neurotransmitters. In central nervous system, in different regions, you need very special concentrations of neurotransmitters. Somewhere for proper function of the brain, at specific time you need glutamate. In other area you need maybe norepinephrine. Still some other area you need serotonin and dopamine, right? And if there was no barrier between the blood and the central nervous system, these neurons will, so these neurotransmitters, very, very precious chemical substances, will drain into blood and flushed away into general circulation. And how brain will keep the concentration of such neurons at appropriate level for functional purposes. So blood-brain barrier also helps the central nervous system to hold the neurotransmitters within the central nervous system. So they are not flushed away through circulation. You know how much blood every minute passes through your central nervous system? Yes. Your brain looks quite active. Yes. Yes. You are almost right. Actually, about 15% of the cardiac output passes from the central nervous system. You know, cardiac output usually in a resting person, adult, is about 5 liters per minute, right? So you can say about 700 to 800 ml blood. You are almost right. 700 to 8 ml blood, right? And when you are exercising, of course, cardiac output improves. Sometimes during exercise, even more than 1 liter blood passes through the central nervous system per minute. And if there is no barrier, neurotransmitter will be flushed away, washed away. Do you think we will be happy man then? Or happy woman even? No. So what are the functions of blood-brain barrier? I have not yet told you what is blood-brain barrier exactly. I just told you hypothetically there is something which prevents the free movement of certain substances between the peripheral blood and the central nervous system environment. And that something is called blood-brain barrier. We'll discuss exactly what it is. But functions of this blood-brain barrier are very important. It provides a stable micro and molecular environment and biochemical environment to the neurons. It provides protection to the neuron, central nervous system, and it helps the central nervous system to hold the neurotransmitters concentration. Am I clear? But now we see exactly what is blood-brain barrier, right? So we'll go to discuss that. The structural details of the structure of blood-brain barrier. Let's suppose here it happens to be your brain, right? It is just for teaching purpose. It's out of proportion, this diagram, but still. Let's suppose here is your general circulation, right? And this is the blood vessel which is passing through central nervous system. And I will enlarge it so that you can understand it more clearly. That blood is going, of course, from general circulation through, carotid system and vertebral arterial system to the central nervous system, and eventually arteries go into arterioles and then eventually into capillaries, right? And then they drain back from the central nervous system through the venous channel back to the general circulation, right? So from here it comes out. Now, the concept is that between Blood, here is the blood, and here is what? Central nervous system, extracellular environment. Between the blood and central nervous system, there is a barrier, and that barrier is here, right? Now we're going to detail what it is, right? Let's put some neurons here, right? These are the neurons, right? They are doing a lot of talking there, as you know, right? So communicating, synapses and all those activities, right, going on here. And it should be very intimate environment. And all the adventurous activity here should not disturb it significantly. The system should be so wise that the things which are really needed by the central nervous system should be able to transfer, and which are not required should not be able to transfer, right? And here is the barrier. Now what is this barrier? It's not a red line there. Let me tell you what it is. Actually, blood-brain barrier has multiple components, right? First I will tell you simple component. We should put a comparison between capillaries in other tissues and capillaries in the brain, right? Because these are the endothelial cells of the capillaries which provide the major function of the barrier. Now, let's suppose we put a capillary. How many types of capillaries are there? There are continuous capillaries, there are fenestrated capillaries, and there are sinusoidal capillaries. Now, continuous capillaries are those capillaries, look here, where endothelial cells don't have junctions between them. Like these are the endothelial cells, and there are not, between endothelial area, there is not significant gap for the fluids to move. So this is continuous capillary, and it is having, what is this? Basal lamina or basement membrane, which is also continuous, right? Then there are fenestrated capillaries. Now, what is the difference in fenestrated capillaries? Fenestrated capillary is that the real difference in continuous capillary and fenestrated capillary is between the endothelial cell structure. What is the structure? That these endothelial cells are fenestrated. These endothelial cells here, these are fenestrated. I will explain what is fenestrated, right? Fenestrated. Now what is fenestration? Fenestration means window. Window. Look at this wall. In this wall, let's suppose this wall is an endothelial cell. This wall is endothelial cell. And if there are round windows here, round windows, these round windows or holes, these holes or pores or round windows are called fenestrations. What are they called? Fenestrations. So actually these endothelial cells, they are having special type of, you can say, holes here, and through these holes certain things can pass very easily, and these are called endothelial windows. We can also call them endothelial windows, or we can call them endothelial fenestrations, right? Now these fenestrations in some areas are really true windows, for example in glomerular capillaries. But in most of other fenestrated capillaries, in the window there is a curtain. There is a curtain. There is a little diaphragm, right? So both of them are anyway called, either there is only window or there is a window with curtain. In both conditions, if any endothelial cell has so many round windows through which substances can easily pass, then we say these endothelial cells are fenestrated. And if there are endothelial cells are fenestrated, then we say these are fenestrated capillaries. But these continuous capillaries, their endothelial cells don't have these pores, or they don't have endothelial windows, or they don't have the, what is this thing called, diaphragm with the endothelial windows. So we say these are continuous. So what is this? What is the thing about the continuous, or other name is continuous or tight capillaries, right? That their endothelial cells do not have fenestrations, and interendothelial cell gaps are also not there, right? Classically, continuous capillaries are found in the central nervous system, an endoneuron of peripheral nerve, I mean brain and the spinal cord and nerves, and continuous capillaries are also found in Yes? Continuous capillaries are also found in, yes, my friend? You like to hide your knowledge, huh? Yes. You don't like to show off. Continuous capillaries are at many places, but at least you should remember muscles. Right? Muscles also have continuous capillaries. I mean, muscles have capillaries where there are endothelial cells without fenestrations. And the muscles, endothelial cells are without fenestration. But in muscle, interendothelial gaps are little bit there. But in central nervous system, right, these continuous capillaries do not have interendothelial gaps. Is that right? These are fenestrated capillaries, right? Then there are sinusoidal capillaries. Sinusoidal capillaries are where endothelial cells have lot of gaps, and somewhere there are really deficient wall of the capillaries, right? Classically, we can say it is present in the liver and the spleen, right? In the liver and the spleen, right? And there are some reticuloendothelial cells also there, macrophages and other. But I will not go into detail. What really I wanted to tell you, the body has three types of capillaries. There are continuous capillaries, there are fenestrated capillaries, and there are sinusoidal capillaries. Continuous capillaries have endothelial cells which are not fenestrated. Fenestrated capillaries have endothelial cells with multiple endothelial windows called fenestrated capillaries, and sinusoidal cells are really broken capillaries, right? Now, yes, please, you have a question.
The basement membrane is continuous in the...
Here it is continuous, here it is a little bit broken, here it is really big in patches, right? Now, we come to the endothelial cells, characteristics of the endothelial cells lining the capillaries of central nervous system, right? Let's come here. Now, if I draw endothelial cell here, this is endothelial cell, right? And this is one more endothelial cell, and here is one more endothelial cell. Now, the first thing, these endothelial cells have a very special thing, because this is a private area. Women like to be behind the curtains, and usually curtains also they like to stitch them more and more. So what happened? There's special stitches here. There's special protein which stitch the, what is this? Interendothelial gaps. These gaps which are between the endothelial cell, actually there are no gap. They are very tightly stitched, right? There are special type of threads. What are those threads? Those threads are basically special proteins called occludins and claudins. Occludins, claudins, and ZO1 and many others. You don't need to remember all of them. But you must remember that endothelial cell, which are lining the capillaries in central nervous system, right? These endothelial cells are very tightly stitched with each other, and substances are not allowed to freely move in between the endothelial cell. Is that right? And this point where stitching is done, this is called, there are junctions between the one endothelial cell and other endothelial cell. There is a junction, but because junction is stitched, it becomes a tight junction. So we call them tight junctions. The classical most important thing which is present in central nervous system vasculature, which act as a blood-brain barrier, is the tight junctions. Is that right? This is the main reason why everything from the blood cannot jump into brain, and everything from the brain cannot jump into here, right? So what is the site? Number one is tight junctions. So let me now make this capillary little bit larger, that there are tight junctions, and I will draw the capillary here, right? This is central nervous system capillary, and here is your, what is this? Endothelial cell. Here is another endothelial cell, and so on and so forth. And here are the stitching done. And what are these stitchings? Tight junctions, and they are made of special proteins, and these proteins are occludins and claudins, and ZO1 and some other also, right? And that is why at least one route through which usually passage occur is blocked. Now, if I draw here I'm not making central nervous system protein, I'm making another type of Now, in this side, this is another capillary from somewhere else in the body. I'm comparing capillary from other part of the body with the capillaries in the central nervous system. This comparison. Now here, number one, these stitches are not there. If these stitches are not there, then what will happen? Substances will be able to move through this. And if substances can move through this, right, this is called larger molecule or polar molecules. Polar molecules are those molecules which are highly charged, horny molecules, charged molecules, right? Or the molecules which are, you can say, water soluble molecule, right? If someone is very charged, dip him into water, right? So what I'm talking about, that these molecules which are larger, which are charged, which are, we say, water soluble, right? These molecules can easily pass through interendothelial gaps, and this is called paracellular route. What is this? Paracellular route of exchange between the vascular system and interstitial fluid. But in central nervous system, this paracellular route is effectively blocked. It's very effectively blocked by tight junctions. This is the first most important thing. If you don't remember anything about blood-brain barrier, just remember one thing. The major reason of this barrier between the blood and the brain is what? Tight junctions in endothelial cells lining the cerebral capillaries, the central nervous system capillaries. This is only one, but story doesn't finish here. You know, this is a very discrete area. Extra protections are there. Actually, blood-brain barrier is a sort of neuroprotective layer, protecting the central nervous system, private environment. Now, there are other reasons. There's another route through which substances pass from the blood extracellular area and in the blood. And that route in other capillaries is through the cells. That is through the cell. When substance passes through the cell, it is called transcellular route. And this is paracellular route. Now in transcellular route, there are many ways. One of the ways that endothelial cell, let's suppose this is an endothelial cell, transcellular, how it can do, that it can make a little, what is this? Depression, and substance which need to be transferred, that will be taken into this pit. And then this will move a little inward, and now substance is in a vesicle, and then this vesicle may fuse on the other side, and this substance will release out. So from here it entered, there it went out, but it passed through a vesicular system. Many epithelial cells, right, which endothelial cells in the many tissues, they are having special type of transport from the endothelial cell from one side to other side. That special vesicle are formed on one side, right, depressions in which proteins or larger molecules which are supposed to be transported are trapped or attached, and then this vesicle, these pits or depressions, they become deeper, convert into vesicle, and vesicle are transported to the other side of the endothelial cell until vesicle fuse with the cell membrane on the other side, and its content are thrown out. And this is called transcytotic vesicular transport. What is it called? Transcytotic vesicular transport. Is that right? And here, do you think such kind of transcytotic vesicular transport should be very effective in these cells? No. Just ask any women. This should not be like this, that men sit in the, what is his, drums and pass in. So this drum business should not be there. So this is another important reason. Why blood-brain barrier is there? Not only because paracellular route is blocked by the tight junctions, but also in central nervous system, endothelial cell show very little vesicular transport. But other capillaries show a lot of transcellular transport through the vesicular system. This is the second reason. Then there is another thing. And what is that? Of course, I talked about that, fenestrations. Here, for transcellular route, not only you can say these are special type of drums which capture the substances and transport to the other side. There were special windows here. You remember endothelial windows, right? And these endothelial window, few things can jump from this fenestrations. And here there are no fenestrations, right? So really the most important thing as a blood-brain barrier right? As a doctor, you must know it very clearly, because in some diseases like infections or like tumors in the brain, or like very high blood pressure crisis, or like infarctions, ischemia, blood-brain barrier is broken. And you must know what's going on in the brain of the patient. But anyway, so what we can say, number one reason for blood-brain barrier is, what is this? Tight junctions. Number two, no fenestrations. No fenestrations. Number three, very poor transcellular, transcytotic vesicular transport. Am I clear? So this is how these endothelial cells are different from others. And even that, yet it is not enough. Now we can say, these nasty men who will love to go in, they are not provided with paracellular route, they are not provided to squeeze in between, they are not allowed to go through windows because there are no windows, and even transcytotic route is very, very special and very limited, so very little chance. But still, men are men. Some nasty men may jump in. Sometimes a neurotoxic substance which is little lipid soluble, right? Neurotoxic substance which is really lipid soluble, somehow it dissolves into membrane and go here. What will happen? These ladies will be alarmed. Central nervous system cells will be very disturbed. So what happens? There are special, very strong guards there. We use the energy, capture these men and throw them out. Right? These are here. These are very special transporters. Right? How they were discovered? Initially, it was discovered by doctors that many drugs are somewhat lipid soluble. They do cross the endothelial lining, but somehow their concentration in the brain is not achieved well. Now they came to know there are special transporters which are present in endothelial cell luminal side, and these transporters, they have a survival value. These transporters will take multiple drugs and throw them out in bundles to protect this area. Central nervous system knows how to protect itself. Is that right? But originally, these transporters were not made by nature for the drugs. They were originally made for lipid soluble neurotoxins. That when somehow in animal's body, lipid soluble neurotoxins enter, right? Even if these neurotoxins get some amount, to some degree, they get into central nervous system, there are special active transporter here, energy dependent, ATP dependent transporters, which will keep on actively throwing them out, right? These are called, yes, what they are called? P, yes, they are pissed off. P, what? Glycoproteins. Glycoproteins. We really call them multi-drug specific, non-specific, multi-drug, non-specific, multi-drug, non-specific active transporters which throw the substances from the central nervous system back to the blood, right? It is good, it's a protective mechanism. But these days, researchers are trying to exploit them, because many of the drugs which we give, anti-cancer drugs, or some strong analgesics, or some strong antibiotics, sometimes we need to get these drugs into central nervous system. But these nasty, what? Very bad guards, they keep on throwing their drugs out. But we really want them there. So now they are trying to find the substances which can block these pumps, so that men can sneak in and they are not thrown out in a ruthless fashion and in a disrespectful way. You understand it? So this is the real site of blood-brain barrier, the endothelial lining of central nervous system, blood-brain barrier. How many things? Number one is, what is this? Tight junctions. Number two is no fenestrations. When tight junctions are there, no paracellular transport. Then, what is there? No fenestration. Then, no transcytotic transport, or very little. And then, it's really unfair, by the way, that first you don't let them go in by paracellular route, then don't let them go through the transcellular route, and in the end, if someone goes in, you kick the ass and get it out. Right? It's really unfair, but it is there. This is how life is. So these are P glycoproteins which keep on kicking them out. All these things help to keep the very private and very secluded and very discrete environment for the neuron very stable, very protected, and working without interruptions and threats. Is that right? But it is not enough. Then there is more component to blood-brain barrier. Even the most important are these. We have talked about 4 components in endothelial cell, right? Tight junctions, no fenestration, no transcytotic vesicles or very little, and P glycoprotein. Then there is a basement membrane here. The basement membrane which is here, this is continuous, right? This is continuous. And yet central nervous system is not satisfied. This ladies put some of very good friends here to watch out that these stitches are working well or not. Right? They send some friends here, and those friends are sitting here and they're looking. These, what are these? Tight junctions are there or not? And if they are somewhere destroyed or they're poor, though their friends give order to produce more proteins, occludin and claudin and Z01 and others, to tighten the tight junctions. Is that right? Now what are those friends? These friends are basically special type of cells, and these cells are called astrocytes. And astrocytes have their foot processes, and these foot processes, right? These are the astrocytes. These are glial cells, and they have these foot processes, and these foot processes are, astrocytes have many foot processes, you know. You can say everywhere. They have many functions, but I will not discuss them into detail. But astrocytes which are near the capillaries, their foot processes approach the capillaries, and these foot processes are slapped against the what? Basement membrane. Right? But remember, they do not provide themselves a real barrier. You know why? Because same substance which is injected in central nervous system, it cannot enter. It will block here. Cannot go beyond. But same substances, if they are injected into central nervous system, they will go through, they will pass in between the foot processes, they will pass the basement membrane, and they will stop here. So it means on this side, they are not very effective barrier. But then what is the purpose? The purpose is that these astrocytes release the soluble substances which force the endothelial cell to produce and synthesize more and more occludins and claudins and Z01 and other proteins, and they become tight and tight. They become tighter. Right? So purpose of these foot processes really not as a physical barrier, because 100% of the capillary lining is not covered by the foot processes. About 80 to 90% is covered. The real purpose is they induce the formation of tight junctions. Right? And how we know that? There are so many ways to know it. One of the clinical points here is that when in central nervous system tumor develop, let's suppose a rapidly growing cancer is there in central nervous system. Those cancer cells develop their own capillaries. And as those cancer cells don't have astrocyte, so capillaries which are within the cancer, let's suppose here is the central nervous system, and unfortunately here the cancer developed, and capillaries which are passing through this cancer area, right? Because there are no astrocyte, these capillaries will not develop tight junctions, right? And in the cancer cells, there will be more exchange and more transport of larger molecules, right? So it can help in diagnosis of the cancer, that radiologists give radio labeled substances which, if there is no cancer there, then those substances cannot cross the blood-brain barrier. But at some point, heavily that substance crosses the blood-brain barrier, it knows that blood-brain barrier is broken there. Am I right? So pericytes, what is the real role of, astrocyte foot processes to induce the tight junctions, to maintain the endothelial barrier healthy. Then another thing, in between this basement membrane, there are special cells which are called pericyte. Right, we will not go into detail of that. They are not playing any special role in this, but they are there. So ultrastructurally, we can say that endothelial cells which are non-fenestrated and with tight junctions and without transcytotic vesicular system, at the top they are very ruthless and disrespectful to the arrogant molecules and kick them out through energy-dependent pathways of P glycoproteins. With that, there is continuous basement membrane or basal lamina with some pericytes, and outside of then beyond that, on the brain side, there is foot processes by the astrocytes, which specially look after that tight junctions are mature and enough tight. So this is the basic structure of blood-brain barrier. Actually, similarly, there is blood-spinal cord barrier, and not astrocytes are not there, but even in nerves, peripheral nerves, in the endoneurium there are capillaries. Capillaries in the endoneurium of the peripheral nerves also have endothelial cell with tight junctions and without fenestrations and without transcytotic, effective transcytotic transport, but not those things. Anyway, now the question is that still there are some things which are required here. Then how these substances pass? Right? Now I'll go into detail what substances normally can pass easily and what cannot pass easily. Right? So let's come to this area and we'll talk about it. What can pass and what cannot pass. Number one, big guys cannot pass. Big guys cannot pass. Right? If molecule is very large, it cannot pass through this. Right? Big molecules, large molecules like plasma proteins do not cross blood-brain barrier effectively. Plasma proteins. And if plasma proteins do not cross blood-brain barrier, it means another thing. You know what is that? All substances which are bound with the plasma protein cannot pass through as well. For example, is that clear? All substances which are bound, or all the drugs which are bound with plasma protein, they cannot enter into central nervous system. Then not only big guys, yes, horny guys, highly charged molecules, right? They are also not, they are not allowed to pass, because when a molecule becomes more and more polar or more charged or become more and more water soluble, it becomes less lipid soluble, and if it is less lipid soluble, it cannot go through this biological membrane. So the guys which are very big guys not allowed, maybe little children, yes, we'll see later, right? Babies are allowed in women's area. So what we are talking about, that big guys are not allowed, right, guys? And then what are these guys? Highly charged guys are not allowed, horny guys are not allowed, and then of course toxic guys are not allowed, even if they are small and even if they are showing as if they are not horny, right? If there's a toxic guy, very dangerous guy, and even if it is small and it's not very charged and it is lipid soluble, it will be actively thrown out if it is not required. So which guys should not be allowed there freely? Big molecules, big guys, horny guys, and toxic guys. Is that right? And you know why and how they will. Then who can go in? Of course, babies. Yes, he knows. There's a baby here and he knows. All right? Babies are allowed freely in the ladies' room. I know you are very jealous of them, but anyway, shouldn't be. Oh, very bad, you know. So anyway, the little molecules, little happy molecules can cross easily, because little molecules are, what is this? Lipid soluble. Very small molecules can very easily cross. For example, water molecules, oxygen, carbon dioxide, they can very easily cross. Carbon dioxide can easily cross either way, but protons not. Protons are the horny guys. They are highly charged. Is that right? Then, yeah, here, one way is glucose. From glucose I remember something. I have my one frustrated friend. That frustrated friend, sometimes we sit in evening coffee and he always talking the same thing. You know, I feel there is a psychological barrier between me and my girlfriend. And every week his girlfriend changes. And he says, my first meeting is usually the last meeting, I don't know why. He's really frustrated. And once he told Dr. Najeeb, please tell me, what is the real reason I cannot get across this psychological barrier or emotional barrier? I try to impress, I try to press, I say, there you are mistaken. Right? Whatever I do, but somehow the girl who is interested in me is usually interested until she sees me first time. So I told him, you have issues. Girls like big guys, but not too big guys. Not intimidating guys. Girls like horny, but not, what I should say, horny from the very beginning. Maybe they can convert carbon dioxide here and then make it polar with water. hydrogen and bicarb. But they don't like that on the first meeting, man is very horny. Horny men are not welcome on like this way on the first date. You understand why? Because women believe that the sex should come after the connection, emotional connection, psychological connection. They need connection and then sex or other things. But my frustrated guy say, oh my friend, he was telling me, what you are telling Dr. Najeeb, women need first connection and then sex. He said he believes sex is the connection. And I said, then it's right. You will have a lot of barriers. You get it or not? So what happens? Big guys, horny guys, he is a big ego. First of all, he's a big guy, too big guy to intimidate. Secondly, all the time horny. Right? Then, other problem with him, he's very toxic also. Whenever he'll sit with girlfriend, if he get a chance, he will talk about all other girlfriends who just dumped him. It's a very toxic attitude. You need to tell her she is the greatest woman in the world. But he is telling always that I'm fearful that you will also leave me the way other did. And she does. You understand? So, but who are the guys who get in? Sweet guys. Later on, whatever happened to them? Glucose is wanted here. Glucose is wanted here. But let me tell you one thing. Neurons like ladies, mainly most of their energy comes from sweet glucose. Neurons, more than 90% of their energy source, right? They use the nutrient for energy, glucose. It should be sweet. Sweet guy. Now, the problem is that glucose is also hexose. It's a larger molecule. Glucose is also larger molecule and also polar molecule. But it's very sweet. So, central nervous system, women will make some arrangement here so that it can go in. You understand? Special facilitated transport for specific molecules. You understand it? I think all of you understand. You are almost adult, right? So what happened? That glucose is needed there, so there should be very special transporters for glucose, right? And if I show this molecule or endothelial cell, this is if central nervous system, capillary endothelial cell, I take this cell here. There are special transporters here, and when glucose bind them, they turn the side and throw the glucose in. Then glucose will bind on this side and this will flip and throw it out. These are special glucose transporter proteins. There are many types of glucose transporters. The glucose transporters which are present on these capillaries, they are called GLUT1. Of course, number one. Do you know why? Why? Number one glucose transporter, because it is independent of insulin. Many glucose transporters are dependent on insulin. But when women really like someone, they don't need they somehow manage it. You get it or not? So this transport of glucose is not insulin dependent. You know, glucose transport and let's suppose in muscles is insulin dependent, or adipose cell is insulin dependent. But here, because they need the sweet guys too much, right? They just plant what is there. This blood-brain barrier still have special molecules which act as a glucose transporters, and these transporters just get a sweet guy and throw it in, right? These are called GLUT1, right? Or glucose transporters type 1, right? What is very special thing? You can write one as like this. You should remember, independent of insulin. Why it is independent of insulin? Because insulin levels in the blood fluctuate a lot. But neurons need a constant supply of glucose. So it cannot be, the glucose transport to the central nervous system should not be at the mercy of fluctuating level of insulin. So insulin independent glucose transporters are there. Am I clear? Then there is another, amino acids are also required there. So as you know, what they will do, they will put a special system for transport of amino acids. So whatever is required in central nervous system, right, very small and lipid soluble molecules simply diffuse in, or from here they may diffuse out. But larger molecule, if they are really needed by central nervous system, there are special carrier mediated transport mechanisms, like I told you, glucose transporter. Then there are special transporters here which are amino acid transporters. Amino acids are required in the central nervous system For neutral amino acids as well as basic amino acids and acidic amino acids, there are special transport mechanisms. This transport mechanism reminds me one thing, that one of the amino acid transport mechanism can transport a drug called levodopa. There's a disease called Parkinsonism, and in Parkinsonism what really happens, substantia nigra, the neurons which are dopaminergic neurons which project to the basal ganglia corpus striatum, these neurons degenerate, and when these dopaminergic neurons degenerate in substantia nigra, right, then dopamine concentration in the basal ganglia or corpus striatum become less, and that produces a lot of you can say motor movement abnormalities. I will not go into detail now, but one way to correct the situation is, because there is biochemical deficiency of dopamine, get the dopamine here. But the problem is that if you inject the dopamine in the blood, dopamine is highly polar and horny molecule. It cannot cross. But there is a precursor of dopamine, that is levodopa. It is also horny to some extent, but sometimes you can fool the system. Levodopa bind with one amino acid transporters, and then it is transported in. And once it is in, then these neurons will convert levodopa into dopamine. Is that right? So glucose transport is special carrier. What is this? What we call it? Carrier mediated facilitated transport. Then there is transport also for the amino acids, glucose, nucleosides, even choline, right? Even thyroid hormone. Thyroid hormone is in two forms, protein bound and free form. Free form can go through special carriers. Then steroid hormones. Steroid hormones are protein bound and free form. Free form of steroid hormone can diffuse in, but protein bound cannot. Right? So what I'm trying to say, blood-brain barrier is very, very, I must say, clever. It knows what to get in and what not to get in, and how to get in. Right? For getting in, simple diffusion for small and lipid-soluble molecules, or carrier-mediated transport classically for glucose and amino acids, choline and nucleosides, right? And some other substances which are really required actively can be transported also, right? Now we come to, this is something about blood-brain barrier. And I was talking about, there's another barrier which is called blood-CSF barrier. And what is the difference? Let me tell you. What is CSF? Cerebrospinal fluid. Cerebrospinal fluid is present, produced by special capillaries called choroid plexus. Capillaries in the choroid plexus actually produce the cerebrospinal fluid in the ventricular system of the brain. Now let me show a ventricle here. Now I'm really going to show that, make your concept clear about what is the difference in blood-brain barrier and blood-CSF barrier. So I was telling, right, here was, let's suppose, this is brain substance, right? This is brain substance. And let's suppose here is a ventricle, right? Ventricular system, right? This is just a simple diagram. Now, the barrier between this area, this barrier was what? Blood-brain barrier. And the barrier, everything which is present in blood does not go to CSF. So there is another barrier here which is called blood-CSF barrier. Now let me go into detail of blood-CSF barrier. Basically, here there are capillaries, and these capillaries have endothelial cell, but these endothelial cells are fenestrated. Now you see the difference. These endothelial cells are fenestrated. These were non-fenestrated. And here are the tight junctions. Here are no tight junctions. Let me compare blood-brain barrier capillaries and blood-CSF barrier system. Now, how do you differentiate? Here, what is the real difference? Here, the endothelial cells which are not non-fenestrated. Here, in choroid capillaries, endothelial cells are fenestrated. And here there are tight junctions, and here there are No tight junction. It means here can be transcellular transport as well as paracellular transport. It means this is the way to get to the ladies system? Answer is no. They have put a barrier but little ahead, just to frustrate you more. You know what they have done? Under this endothelial cell, of course, what should be there? Basement membrane. Under that, there are cells. Here, what are these? Ependymal cells. What are these? Ependymal cells which line the ventricular system inside. These ependymal cells, when they come over these capillaries, they become very specialized ependymal cells which are lining the ventricle. And these modified ependymal cells are called choroidal capillaries. Epithelial cell. Choroidal epithelial cell. Right? Again, let me tell you. Blood vessel, first of all, you must know, this is a ventricular cavity. Ventricular cavity, especially lateral ventricles, third ventricle and fourth ventricle, all of them are having special structures which are called choroid plexus. Choroid plexus have special capillaries on one side. There is the endothelial cells of capillaries in choroid plexus. Then there is basement membrane. Okay, let me tell you. There are endothelial cells. Then there is basement membrane of endothelial cell. Then there is basement membrane of ependymal cell. And then there is these modified ependymal cells which are called choroidal epithelial cells. You get it? Anything which has to go from here, it has to go like that. Right? Actually here, in blood-brain barrier, tight junctions were between what? Endothelial cells. Right? Here the tight junctions are between choroidal epithelial cells. That's a little trick. But actually still they are very self-protective and very, very discreet. Who comes in and who doesn't. Is that right? So what we can say, what is the difference between blood-brain barrier and blood-CSF barrier? As we'll say, protein-bound substances and many substances cannot freely pass from the blood to the brain extracellular environment, or many substances cannot pass freely from the blood to the cerebrospinal fluid through the choroid plexus. Here, why they cannot pass, we already discussed, due to non-fenestrated capillaries and tight junctions in the endothelial cells. Here, they do pass through because endothelial cells are fenestrated, so many substances do pass. And there are interendothelial gap also, not tight junction, still many substances do pass. But frustration is waiting for them. As soon as they go deeper, they go pass through endothelial layer, they do pass through the basement membrane, even going around the pericytes. But when they reach the epithelial cells, choroidal epithelial cell, on their apical sides, they are having stitching. And that stitching or that tight junctions do not allow the substances from the blood to freely move to the cerebrospinal fluid. Am I clear? So we can say most of the substances in the blood cannot freely enter into central nervous system or into the brain and CSF. So we can say there is blood-brain barrier and blood-CSF barrier. Do you have any question up to this? Right. Now we talked about basically structure of the blood-brain barrier. We have also talked about structure of the blood-CSF barrier. We also know that what are the stoppers here and what are the promoters for the transport, right? And after this, I will tell you under which circumstances blood-brain barrier is pathologically broken down. Of course, there are so many ladies there. Sometimes these walls are broken down, right? Sometimes there can be tragedy. There can be some sort of aggression, right? Now, under what circumstances blood-brain barrier can be broken? Of course, those are pathological conditions. Let me tell you, women are very clever. They have kept some special secret windows. In the blood-brain barrier, there are some secret windows. They are physiological windows, right? Those windows are present around the ventricular system. Those windows are present around the ventricular system. Those windows are called, those areas with the windows through where many substances can jump in or jump out. Sometimes ladies do jump out also. Not neurons, but their products. Now, what happens that neuronal system is keeping some very special and secret what? Windows, right? Through which special exchanges are done for special purposes. I will not go into the detail of purposes right now, but later on. And these windows are usually around the ventricular system. And these windows are called circumventricular organs. What are they called? Circumventricular organs. These are physiologically present, normally present. This is the way to get in respectable way, not trying to pushing everywhere. Right? There are what? Circumventricular organs. Those circumventricular organs I will discuss after the break. They are physiological leaking point or physiological breaks or normal breaks in blood-brain barriers in some specialized areas of central nervous system. Circumventricular organs. We'll discuss that after the break. But before going to the break, I would love to mention that sometimes pathologically blood-brain barrier is broken in multiple places. Why? Why the blood-brain barrier may be broken? Number one, it may be trauma, severe head injuries, brain injury, or it may be inflammation, infection, right? It may be irradiation, it may be neoplasm, it may be hypertensive crisis, very high blood pressure. We call it hypertensive encephalopathy. So in all these conditions, blood-brain barrier break at multiple points, right? So if someone is having severe trauma to the brain, blood-brain barrier may break. These tight junctions may break, right? In severe inflammations or infections or irradiations or neoplasms, if they are present in some part of the brain, there may be broken blood-brain barrier or hypertensive encephalopathy. And what happens when blood-brain barrier break down? You know it that very well. What will happen? That many substances will inappropriately enter in, right? Am I clear? And when those substances will enter into central nervous system, water will follow. And swelling of the brain will occur, or that part of the brain will occur, and we say there is edema of the brain. And this edema of the brain is due to vascular problem. So we call it vasogenic edema. Which edema? Vasogenic edema. So what is vasogenic edema of the central nervous system or brain? Vasogenic edema is when due to some pathological process, blood-brain barrier is disrupted in some part of the central nervous system. Either endothelial cells are destroyed or interendothelial tight junctions are disrupted, and then lot of solutes from the blood and fluids may enter into central nervous system and brain may swell up. It may be a life-threatening situation because when brain swell up, it is in a tight, limited cavity of the skull, and when it swell up, right, it will press itself against the skull structures and there will be raised intracranial pressure, and that can lead to death and many complications, right? So this is one problem. Then another clinical point which I want to mention, that in newborn, blood-brain barrier is there, but it's not as effective as adult. So in newborn, if some, bilirubin, which is the breakdown product of hemoglobin, bilirubin is highly neurotoxic substance. Normally in healthy person, bilirubin is not allowed to enter in central nervous system, right? Because it is neurotoxic. But in babies, number one, blood-brain barrier is not very effective. Number two, sometimes in newborn, bilirubin, unconjugated or free level of bilirubin goes very high. There are multiple reasons. Number one, in the baby, fetal red blood cells are breaking down and being replaced by mature blood cells or adult type of hemoglobin, and there's more breakdown of hemoglobin in babies, produces more bilirubin. Secondly, in babies, in the liver, enzyme which conjugate the bilirubin, glucuronyl transferase, that may be immature or less glucuronyl transferase. So conjugation system of what? Free bilirubin conjugating with the, binding with the glucuronic acid and making it highly polar so it can flush out through urine, making it water soluble. This system may not be effective. So in newborn, if due to some reason bilirubin level goes very high, right, then a very high level of unconjugated bilirubin can cross the blood-brain barrier. Blood-brain barrier cannot manage such high concentration of, and in babies this may lead to the yellow coloring of basal ganglia and cerebral cortex, and even in very high concentration basal ganglia may be damaged. That may lead to dysfunction, central nervous system, neurological dysfunction. That condition is called kernicterus. What is it called? Kernicterus. right? So these were some points related with the blood-brain barrier, right? And after this discussion, we will have a break, and after the break, we will discuss about that very special secret tunnels and windows which ladies are keeping there, central nervous system is keeping there for the special welcome and jumping in and jumping out, and those windows are called what? Circumventricular organs. What are they called? Circumventricular organs, right? That we'll discuss after the break. Last point before I go, that going to very high altitude, going to very, very high altitude can also produce damage to the blood-brain barrier. The reason being, when you go at very high altitude, oxygen concentration become very low, and that hypoxia can damage the blood-brain barrier. How? When there is severe hypoxia, the mitochondria and endothelial cell cannot work well, and many active transport mechanisms fail, and blood-brain barrier does not remain effective. So patient may develop cerebral edema when people go to, some people when they reach to very high altitude. That is also a type of vasogenic edema. Yes, you have a question.
Sir, what is cytotoxic edema?
Cytotoxic edema is different. That is not related with this topic. We'll discuss in neuromedicine lecture. Any more question? Okay, let's have a break.